All right, now let's talk about smooth muscle. So I talked about it before, but smooth muscle, I guess I'll just draw a smooth muscle cell. It doesn't have myofibrils. It only has, uh, it has an array of these intermediate filaments, right? Okay, so I'll just kind of make it look like that. I'll only do it for like half the cell just to make things simple. But then there are going to be these dense bodies here. So scatter. I'll only do like 3 just to make it simple. Okay, so there are 3 dense bodies there. Now these dense bodies can either directly or indirectly uh, bind these intermediate filaments to the sarcolemma. Okay. Um, yeah. So maybe the dense body is actually on the sarcolemma, so it's actually. So I'll draw. This is your sarcolemma, and then maybe this is your intermediate filament, and it's sort of bonded to the sarcolemma directly. Indirectly would be like if there was another dense body over here, and then another intermediate filament came over here. This intermediate filament would be indirectly bonded to the sarcolemma. But that's just yeah. Okay. So we've sort of established that there are dense bodies and also intermediate filaments. And there are also going to be thin filaments and thick filaments. Now, these thin filaments and thick filaments are going to be located between dense bodies. So, for example, maybe between these two. I'll just draw them like this. You'll see why in a sec. It will look something like that. Okay. Yeah. So let's zoom up closer, make it easier to see. So this is one dense body. This would be a thin filament. Then this would be a thick filament right here. Actually, make it a little bit more detailed. You might recognize this structure. It is, it's like myosin, right? But instead of what we normally see where it goes like this, right, on the other side, we're going to see that it actually goes like this, okay, in smooth muscle. Okay. I don't know why they look kind of different sizes, but that's not meant to be. That's just my drawing. Anyways, then you would have another thin filament over here. Okay, so this thin filament, is actin. So actin, okay. And then you have 2 of those, right? You have one over here too. And you also have a thick filament, which is myosin. Okay. So you might already be able to guess how these provide this thin and thick filament. These thin filaments and these thick filaments are able to provide a driving force behind, contraction in the cells. Just like myosin and actin work together in cardiac muscle and skeletal muscle, they're able to work together here. So you can imagine if they were able to interact, the myosin heads would push against these actin filaments, and then the actin filaments would sort of come together. Oh, and shoot, there's another. I forgot to draw that. And that will result in contraction. Okay. Forgot to draw this. Okay. Okay. Right, and these are all scattered throughout the cell, so if they were all to contract at the same time, the entire cell would contract. You can imagine. Okay, so how is this going to be regulated here? Because we don't want it to always be contracting, right? If the myosin has access to ATP to push against this thin filament, then it will always be contracting, and we don't want that. We want to be able to control when that happens. So what we can do, or what happens, is there is tropomyosin here, okay? And tropomyosin is also present in myofibrils on the actin, and the tropomyosin is going to block the I'll just draw it like this. Doesn't necessarily look like this, but it's blocking the myosin from interacting with the actin. Now, there isn't actually going to be troponin present, so troponin is not going to be what moves this tropomyosin. If you remember before, it was troponin binds to calcium ions, and then that moves the tropomyosin. That's not going to happen in this case. In this case, what actually moves, or is believed to move, the tropomyosin is phosphorylation of these myosin heads. Okay. Now, how do we regulate the phosphorylation of these myosin heads, right? Okay, so what happens is we have our sarcoplasmic reticulum here, right? And it's going to be containing calcium. Okay, not to scale or anything, just to give you an idea. It's going to have some calcium in it. There is a calmodulin protein, or calmodulin, over here inside the cell. So just to give you an idea, that's not really what it looks like, but kind of. But yeah, so this is not what it really looks like. This is just you know, to give you like a basic sense for what's going on in the big picture. But anyways, this calmodulin, okay, is going to it's going to require calcium. First of all, I won't say what it does yet, but it needs calcium. Okay, now calcium can enter the cytoplasm and interact with this or sarcoplasm in two ways. It can either enter directly through outside, and that occurs when there is depolarization. Okay, then the calcium channels open and calcium will come in. So I'll just draw like some positive charges to represent that. Okay. Yeah. And then the other way is you can have these hormone receptors. Just draw them like this, or one like this, and your hormone will attach to that, and then that will result in a secondary messenger going to your sarcoplasmic reticulum, and that will cause calcium to be released from your sarcoplasmic reticulum. Yeah, and then your calcium, now you have calcium in your sarcoplasm, and it will be able to go with your calmodulin. Okay. Okay. So now you have calcium bonded to this calmodulin. And what this can do is it can activate something called myosin light chain kinase. Okay. Myosin light chain kinase. And let's just say it looks like this, something. Okay. Myosin light chain kinase. And on its own, it's not active, but when it is bonded to this calmodulin and calcium, it is active. Okay. Now, so let's just say they're together now. Select this really quickly. Something like that, maybe. Okay. So they're together. All right. By the way, these things are calciums, just to make that clear, just in case it wasn't clear. And this myosin light chain kinase is going to, along with ATP, so ATP just around the cell, and myosin light chain kinase together will result in this phosphorylation of these myosin heads here. Okay, and that will result in the myosin heads being able to interact with this actin and being able to contract. So it can either be a hormone that's causing it with a secondary receptor, a secondary messenger, or it can be just calcium that's directly going in to the cell because of depolarization. Okay. Yeah, so that pretty much concludes this video. Thank you.